Okay, we're on slide 25, and it happens to be an animation. And I think these BioFlix animations are good. I think they help you to really visualize the inside of the cell and all of the structures, and this, you can see the cytoskeleton and the different filaments. Like this filament right here is very thick. This is a microtubule. The thinnest ones are the microfilaments, and then the ones that are kind of intermediate. Do you see this one? That would be an intermediate filament, because they're in, kind of in the middle as far as the thickness. So anyway, I encourage you to watch it, but if you find that you don't really care for the BioFlix style of animation, you don't have to watch any more after that. But this is one on, it's called A Tour of an Animal Cell. And the BioFlix animations that are being included in the slides are the reason why I'm having to do a PDF of the slides instead of the PowerPoints slides, because it won't upload. It's like too large or something. But I can do a PDF of the slides and then send it to Blackboard as a link. Anyway, you don't care about that. So that's on slide 25. And then there's the tour of a plant cell. So it's good if you watch both of these back to back, you can get a good comparison of an animal versus a plant cell. This organelle right here that's got the little stacks of green disc in it, this is a chloroplast, and that's what the plant uses for photosynthesis. This large organelle inside the plant cell that's kind of a light green shade, but this large one is the central vacuole that stores water. So this is slide 26, and now we're going to move on and talk about the nucleus. All right, so the nucleus of the cell is the control center, and it contains the cell's genes. And it says most of the cell's genes. Don't worry about that. It contains, like, the only other place you have genes inside of a cell is going to be the mitochondria has its own DNA, like it has special mitochondrial DNA. But almost the genes for our traits, all of them, the genes for every trait that we have are located in the nucleus, and they're located in our chromosomes, okay? It says here that the nucleus is the most conspicuous organelle. That means it's the largest, and you can see it the most clearly under a microscope. The membrane that surrounds the nucleus is called the nuclear envelope, and it does have pores, so it allows material to cross through it. And then I'm skipping some of this because you don't really have to know all of it. And like I said, the DNA inside the nucleus is organized into structures called chromosomes, but mostly while the cell is not actively dividing, the DNA is in a form we call chromatin, and it's very, very spread out in the nucleus it's spread out so much you can't see the individual chromosomes. But when the cell starts to divide at the beginning of cell division, chromatin begins to condense and coil up, and it forms rod-shaped structures called chromosomes. So you can say chromatin or chromosomes. It just depends on if the cell is at rest, and then it would be chromatin, or if it's dividing, and then it would be chromosomes. And it tells you that in the next sentence. And then the other structure inside the nucleus is the nucleolus. And the nucleolus synthesizes ribosomes. It makes ribosomes. I know it says ribosomal RNA here, but a ribosome is composed of ribosomal RNA. So basically, the nucleolus makes or synthesizes ribosomes. And then ribosomes synthesize proteins. They are made of ribosomal RNA and protein, and they actually build proteins. Now, you can find ribosomes free in the cytosol. We call those free ribosomes. Or you can find them attached to the ER, the rough ER. They won't be attached to the smooth ER, but they're attached to the rough ER because the rough ER also synthesizes proteins. Okay, so now we're going to talk about the endomembrane system. You may have learned in you like elementary or middle school or even high school, you may have learned that the endomembrane system of a cell is the part of the cell that works like a factory. So that's what we're going to talk about next. And it starts with the nuclear envelope, but it really actually begins with the rough ER, because the rough ER is attached to the nuclear envelope, and then the smooth ER is outside the rough ER. So your endomembrane system will consist of just the outside of the nucleus because the ER is attached to it. So nuclear envelope, the ER, the Golgi, lysosomes, vacuoles, and the plasma membrane. And then smaller vacuoles are called vesicles. So we're going to start with the endoplasmic reticulum in general. Is basically a system of membranous channels that acts like a little factory and produces some kind of product. It may be a protein product, it may be a lipid product, it may be phospholipids to add to the phospholipid bilayer in the plasma membrane. It may be insulin. It may be a cell of your pancreas that makes insulin, and insulin is a protein. So it's a hormone, but it's a protein hormone. So the endoplasmic reticulum functions as a place where products are made, okay? So there are two types. There is the rough ER that has ribosomes, and smooth ER does not have ribosomes. And this is what basically it looks like with the nuclear envelope here. Rough ER is attached to the nuclear envelope, and then the smooth ER is outside of the rough ER. All right, so the functions of the smooth ER are that primarily we think of it as it synthesizes lipids. It does not synthesize proteins because it does not contain ribosomes, but it does synthesize lipids, like steroids, for example, like cholesterol is a lipid, and just different fats, different types of fats. Cholesterol is a lipid, and then all steroid hormones start out as cholesterol, and then they're converted to testosterone or estrogen or whatever. So it could be cholesterol, it could be steroid hormones, just some type of lipid, okay? But another thing the smooth ER does is it plays a role in detoxification. So you find a lot of smooth ER in liver cells because that's one of the roles of the liver. And there's a special type of smooth ER in muscle cells that stores calcium ions. It's called the sarcoplasmic reticulum. So sometimes you find that the smooth ER will store calcium ions. The rough ER has ribosomes attached. So it's going to function in producing proteins and distributing those proteins into transport vesicles. Okay, so what I'm reading says the rough ER has bound ribosomes, which secrete glycoproteins, which are proteins bonded to carbohydrates. But it's not just that. It's not just glycoproteins. Glycoproteins are found in the plasma membrane, and they are part of the glycocalyx, which help. Their function is cell recognition. So basically, if you were to donate an organ to somebody, their tissue, their glycoproteins of their tissues would have to match your glycoproteins, okay? But anyway, glycoproteins are not the only proteins that are made by the rough ER. So it would be glycoproteins, and it would be other proteins, all different types, made by those ribosomes and then modified in the rough ER. And then what happens is the rough ER, when the protein gets to the end of one of those channels, then it can pinch off and become a vesicle. And so we call those transport vesicles, and that will take the protein product to the Golgi. All right, and the Golgi apparatus will then take the products from the rough ER, modify them, and package them into additional transport vesicles, and those vesicles will take them wherever they need to go, either somewhere in the cell or outside of the cell. So here's how it works. Let's say that these little blue vesicles came pinched off from the rough ER. Now they moved here to the Golgi, fuse with the Golgi membrane, and now whatever is inside of those vesicles, whatever protein product is inside, is going to be modified. And then once it's modified, it'll be packaged. And you see how the end of the Golgi can kind of form these swellings and then pinch off. So it will package the product into a vesicle, and then that vesicle is now a transport vesicle, and it will take the product wherever it needs to go. One of the vesicles that's produced by the Golgi and the rough ER is the lysosome. So lysosome is a special vesicle that contains hydrolytic enzymes, and enzymes are proteins. So these hydrolytic enzymes are produced at the rough ER, and they're digestive enzymes. So lysosomes function in digestion. And by the way, plant cells don't have lysosomes. So lysosomes contain digestive enzymes, and these are enzymes that were produced by the rough ER, transported to the Golgi through one of those transport vesicles. And then what a lysosome can now do is, let's say that you have a cell in your digestive tract that has engulfed a food vacuole. Well, it's just a large sac that contains food that you just ate, you know. So a food vacuole is inside of this cell. And so what's going to happen is a lysosome is going to come along and fuse with that food vacuole. And then it's going to empty its enzymes into the food vacuole, and those enzymes are going to break down the food particles into smaller macromolecules, and then into smaller molecules, nutrient molecules, and then they can be absorbed into our blood. So this is what it looks like. Here's the digestive enzymes. Here's the lysosome. Lysosome is purple. Lysosome is fusing with a food vacuole that just basically formed as the food particle was engulfed through phagocytosis. So you have this food vacuole that was formed by phagocytosis. Lysosome fuses with it and breaks down the food inside. Now, another thing that can happen is lysosomes can do the same thing with old, worn-out organelles. So you see this mitochondrion is inside of a vesicle that also contains a peroxisome, because peroxisomes can break down fatty acids, but lysosomes can break down every type of substance. So together, they will break down all of the components that are of that mitochondrion, because it's just going to be broken down and recycled to make a new mitochondria. So another thing that can happen is your white blood cells will phagocytize or engulf bacteria or cells infected with virus, and lysosomes will fuse with those and break down and basically kill the bacteria. And then cells can actually perform cell suicide called apoptosis. Programmed cell suicide is when the lysosome just bursts open inside the cell, and the cell begins to digest itself. Cancer cells will do that sometimes, so they won't spread, you know. So vacuoles are just large vesicles. And we especially have an important vacuole in the plant cell called a central vacuole. Plant cells have a central vacuole for storing water. Contractile vacuoles are found in some protozoans, like the paramecium we saw in the first picture. That paramecium is a type of freshwater protist. So a paramecium has contractile vacuoles because since it lives in fresh water, it's constantly taking on water, and the contractile vacuole will pump out the excess water so it doesn't swell up and burst. Food vacuoles are formed when cells phagocytize or engulf large food particles. Okay, so this is a picture of the central vacuole inside of a plant cell. A plant cell is going to have three structures, by the way, that are different from animal cells. It'll have a central vacuole for storing water that you don't see in animal cells. It'll have a cell wall on the outside, and then it will have chloroplast, and animal cells don't have those. There is an animation on the central vacuole, slide 48, and then a review of the endomembrane system. You see the nuclear envelope, the rough ER attached to the nuclear envelope. The smooth ER is over here attached to the rough ER. Then you have the Golgi. And so the smooth ER produces lipid products, and the rough ER produces protein products, packages them into transport vesicles, sends them to the Golgi. The Golgi packages those products again into transport vesicles after modifying the products. And those transport vesicles can be sent to the plasma membrane and secrete the product outside the cell, or maybe that vesicle becomes a lysosome, and the lysosome stays inside the cell until it's needed to digest something. The endomembrane system, I would encourage you to watch this video if you can. Of all the videos we've seen, I would watch this one, and it's slide 51. And then we're going to talk about mitochondria and chloroplasts just in general. Mitochondria is found in all eukaryotic cells. It is the site of cellular respiration, which is when the oxygen and glucose react to form carbon dioxide and water. But the substance that's generated as a byproduct of cellular respiration is ATP, adenosine triphosphate. And that is the molecule that our cells use for energy. So basically, mitochondria produces energy for the cell. And every eukaryotic cell has mitochondria. So that includes plant cells, fungi, animal cells, and protozoans and algae. All have mitochondria. But only plant cells and algae cells have chloroplast. And so they're only found in plant cells and in algae. And that is where photosynthesis is carried out, which is when sunlight is absorbed by the chloroplast, and it's used to convert carbon dioxide in the atmosphere into glucose, or food for the plant or the algae. So that's where chloroplasts carry out photosynthesis. So animal cells don't have chloroplast, and other cells besides plant and algae don't have chloroplast. And then it does list that peroxisomes are oxidative organelles, but I mean, they basically function in breaking down fatty acids, specifically fatty acids. Now I'm going to skip this on this theory, but there's an animation on the mitochondria, which you can watch in slide 58. And then a little bit on chloroplast. Chloroplasts contain the pigment chlorophyll, which is green, and that's why they are green. And they are found in the leaves of green plants, and they're also found in algae. Chloroplasts are. And a chloroplast consists of stacks of green discs called thylakoids. So the thylakoids are the part of the chloroplast that absorbs sunlight. They're green because they contain the pigment chlorophyll. And then the thylakoids are surrounded by a fluid called stroma. So you can watch an animation on chloroplast in slide 61, and then you can see here, this is a chloroplast where you can see inside of it. And you can see these green discs that are stacked up. They're called, each green disc is a thylakoid. And then outside of all of those green structures is the fluid called stroma. And then over here on the right is this is just a light microscope image of an algae, and it looks to me like an algae we call Spirogyra. And it's just to remind you that algae and plant cells have chloroplast in them. Peroxisomes are primarily involved in breaking down fatty acids. And I don't see that in the notes. So basically, when the peroxisome breaks down fatty acids, it forms, as a byproduct, it forms hydrogen peroxide. And hydrogen peroxide is toxic to our cells. So peroxisomes also have an enzyme their own special enzyme that will break down the hydrogen peroxide so that it won't build up and become toxic. So peroxisomes break down fatty acids. It says it right here. And in the liver, they help detoxify alcohol and other harmful substances. So we also saw that the smooth ER does that as well. So smooth ER and peroxisomes can help detoxify alcohol and other substances in the liver. And all right, we're going to start our next recording with the cytoskeleton of the cell, and we'll talk about the different protein fibers of the cytoskeleton. And this is slide 66.